What's up, y'all? Alright, I did slightly a little bit more work behind the scenes. Um, I fixed this up a little bit. I think this looks nice. Um, added some papers about why we should care about this particular enzyme. This one's actually pretty neat. So I ran an AlphaFold prediction on this guy, and this was like, oh shoot, I probably shouldn't be showing my email. Let me cover this. Uh, my camera just okay. Yeah, give me a second. Do I have it here? Yeah. Okay. All right. So this paper right here outlines like a promiscuous lengthy peptide synthetase. So they're all quite promiscuous, meaning they can accept variable sequence, variable substrate sequence composition and length. But this one's special in that it can accept like I think up to like 79 different ones. And I honestly should read this and kind of like understand how they figured that out. Um, Assuming they'll probably ask me a question on this. If I bring it up, they could ask a question. Is the problem? So you kind of want to be strategic about what you bring up. But also, this would be like more than halfway through the presentation, and they'll probably be testing me more so in the earlier part. So here they'll probably let things slide. Um, but anyway, so I think it's might be in the this yeah. So it's this number of putative prochloricins. I just spilled my drink. Hold up. So I don't know what that is. A large and unusually diverse group of lengthy peptides found in strains of marine cyanobacteria, which are among the most abundant photosynthetic microorganisms on Earth. Hmm. Photosynthetic? Interesting. Cell to cell signaling. Huh. Wait, who did this? Oh, Vanderdonk, of course. Um, so what is this, like NMR? Yeah. LD, LD. And I also need to understand like what these kind of mean. I know what a D and an L amino acid are, but assigning D and L is kind of tricky because of this sulfur here. I'll have 2s, 3s, 6r conformation. The lanthothianines had 2s irrespective of direct cyclizations, indicate that most, if not all, of the rings are formed enzymatically by program lanthothianine synthase and not by non-enzymatic processes as previously suggested. That's pretty interesting, actually. So, I'm trying to see one that has a similar ring A to mine. Leader, as you can see, is very conserved. And this enzyme just goes to town, frankly. All right, but anyway, so like this prediction right here looks like it's in its closed form, which is like super strange. Like usually they have like a very open um, confirmation, but here it's actually looks very closed. So I'm wondering if like I could use this as like a homology model for like these other predictions, you know? But it's strange because I don't understand why it would predict it in the closed form. I could probably try like another seed and see if it's kind of more consistent, just because I'm curious. And here I am getting distracted again. All right, so here's what I want to do basically. So we do need to circle back to this and probably at least read and understand a lot of these. But I really like the key thing here though is like this takes priority. Um, so what we're going to do is like pretty much understand why this is true from these papers. So let's start with the distributive and directional behavior one. So while we can kind of test ourselves. So what I'm going to do is just copy and paste this, give it to ChatGPT, and say, read this paper, explain the experiments in detail, what they found, how they know it's true, and the implications of the results in the context of peptide maturation. All right, let's get out our notes here. I think I've updated this. No. All right, let me black out my screen again. Okay. All right. Okay. So we are going to basically try and summarize this paper. So what experiments did they do? All right. Our bifunctional enzymes use Fourier, so they use Fourier transform mass spectrometry to analyze these activities. In vitro, the dehydration is shown to take place in a distributive mechanism. What does distributive mechanism mean in this context? I think that means it binds and unbinds. With the buildup of intermediates observed by electrospray mass spectra, the cyclization activity was monitored through alkylation of free cysteines and intermediates, providing access to regio selectivity of lithium ring formation. Cyclization process largely anti-Schumann directional of action, forming a 4. Okay. Yep, talks about niacin. Reconstituted in vitro, studies in these enzymes catalyze the dehydration and their peptide substrates generate. Yeah. So they already knew this ahead of time? The timing and order in which these fascinating enzymes carry out these multiple modifications is currently unknown. So that actually is kind of what I want. So we can duplicate this. And then we can do what we do not. Well, we do know the order. Maybe it'd be like reasons. To address these questions, Fourier transform mass spectrometry using methodology analogous to Parsity. However, because cyclization does not result in change in mass, and because the rings are intertwined, answering these questions is technically more demanding, important, and ultimately may help the manipulation of these impressive catalysts for lentibiotic engineering. I like that phrase right there, lentibiotic engineering. Okay. How would I feed this in? So because this enzyme mainly has led us to find some bacteria without modifying the enzyme. With regards to engineering novel lentibiotics. Okay. All right, so kinetic time course of phosphorylation of, so LTCM is just another dehydratase. It's another lantipeptide synthetase. So does it look similar to ours? This is ours, Hallam 2. Not really. This looks more like an M1. prior to the elimination step that may or may not occur in the same active site. In addition, prior studies have demonstrated that relatively high substrate concentrations the LTCA substrate is converted into fourfold dehydrated product without accumulation of intermediates. Interesting. So that means it all happens in one fell swoop. Is that how you say that? Yeah. Oh, we didn't play our Wordle. We're gonna do that in a second. I'm kind of locked in right now. Research suggests that this observation is not to a processive mechanism, the enzyme, but rather a slow deaggregation of peptide substrate followed by faster enzymatic processing. I don't know what that means. Okay, that's weird. At much lower substrate concentrations, partially are observed, but the amounts of materials these conditions are greatly limited. A possible solution to obtaining sufficient amounts of intermediates for investigation is provided by the arginine 399 MET mutant of LTCM that carries out the phosphorylation step but has strongly impaired elimination activity. So this participates in the elimination. Do I have LTCM? That's another reason why this stuff takes so long for me to finish, because like while I'm reading, I'm like researching basically. Where do I want to go? Here. Let's see. No, I do not have LTC. That's weird. Maybe I should grab that then. LTCA. Let's see if you need prod. That's it. LTCA. No, I'm gonna have to look it up. Do they have the accession code? Accession code? They'll probably say it in a minute. That's interesting though. They're like analogous residues in say like Hallam 2. It'd be like just one of these probably. So 399, like it'd probably be something like mine would probably be like 300 here. But we believe that the, oh, or 199, something like that. Buildup of phosphorylated intermediates. The presence of these three cysteines provide a hurdle. Therefore, all three cysteines were replaced with alanines by the resulting mutant peptides will be referred to as cis-free good substrate for LTCM, resulting in the expected 4 dehydrations that were shown in figure 2. After 1 minute of an intermediate product, after 5 minutes, the mixture was corresponding to 1 to phosphorylations by 10 minutes. So 1 minute, there's a single phosphorylation. 5 minutes, a mixture was observed corresponding to 1 and 2 by 10 minutes with 2, and then after 40, this threefold was generated. So these stars indicate the number of phosphorylations there. So the first three can happen without any rings installed, it seems like. And even with a residue phosphorylated. The octuple collisionally activated dissociation did not result in the number of backbone cleavages required. Okay, additionally, due to OCAD being a slow heating fragmentation method, the intact mass of being somewhat low, relevant to intact phosphor. Better fragment coverage. So what they're doing, I think, is breaking up the peptide to see what specific regions have that phosphate installed. Indicates that all modified serines are localized to the propeptide structural region. We note that the fragment ion, the N-terminal his tag in the leader peptide does not correspond. Numbering herein is depicted in fragment 1. Okay. Ambiguous showed that these are located at threonine 52, serine 54. So it says slightly favors serine 52, can phosphorylate that. Okay, they number it. Wait, is it talking about Hallam 2? No. So Hallam 2 is basically to validate what they saw from Hallam 1. Or LTC 1, I guess. LCT 1. Unlike Hallam 2 does generate partially dehydrated intermediates in the presence of relatively high concentration substrates as is shown in the time course in figure 3a. Okay. So that likely means Hallam 2 unbinds a lot of these things, probably. In a distributive manner. Demonstrated the first four were located 61, 62, 67, 71. So boom, 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 boom. So I think the B ions means that it was fragmented from the N terminus. Also confirmed recent collisions based on site-directed mutagenesis experiments that serine 82 escaped dehydration. Yeah, so that's that one there. T77. 2, 3, 4, 5, 6, So it's like this one maybe? I don't know. Let's just number all of them. Okay. 3, 2, 1, 0, 9, 8, 7. So this is 77. Boom, and then boom. Should be noted that dehydrations can be attributed to enzymatic dehydrations, not the result of. Okay. Are not distinguished by mass. Alkylation of all free cysteines present in intermediates was utilized to differentiate cyclized and non-cyclized cysteines. Iodoacetaamide adds. Okay. So with the buildup of intermediates, you can alkylate the cysteine, and if they're free, then that means it hasn't been cyclized yet. Nearly all possible combinations of dehydration and cyclization. Each observed ion can be annotated with respect to the number of dehydrations. Unfortunately, even with this derivatization, significant mixing of fragmentation spectra was observed due to partially overlapping isotopic distributions in such mixture. Okay, I don't know what that means. The combination. The ions containing 6 and 7 dehydrations and between 0 and 4 were analyzed to particular depth. Observation of an unmodified B59 ion localizes all dehydrations as anticipated. Demonstrated a single alkylation was present in serine 82 to cysteine 84. These data suggest that all other cysteines underwent cyclization and therefore that ring D, fragmentation. Okay, I don't get this conclusion here. Therefore, these ions show that one alkylation occurred on the second. It must be that. They always say penultimately. You know, God forbid they say second to last. Finally, fragmentation of peptide product of 6 dehydrations and okay. I think I understood that. Let's see what ChatGPT said. Big picture, what they wanted to know. Enzyme binds once and does all modifications before letting go. You wouldn't see, okay, so processive means the enzyme binds once and does all modifications before letting go. You wouldn't see many partially distributive. the enzyme lets go after each step, intermediates diffuse away and can rebind. You do see ladders of partially, okay. I wonder if there's any example of LAN-M processive mechanism, because that would be nice for me. Yeah, I don't think there's anything. Um, um, lactative for synthesis is highly processive. Yeah, it's just talking about the phosphorylation elimination, which is kind of makes sense. But all right. Okay, so I think we can go ahead and start writing the summary. What experiments did they do? They used high resolution FT Fourier transform, ICR. I don't know what ICR. On PTMs now? Yeah, you can say that. Really, I'm just trying to get some like background knowledge on how we know these things for my enzyme. So I'm going through each of these papers and making sure I understand the experiments they did and how they came to those conclusions. Because my committee is probably going to grill me on that. LTCM phosphorylation, how they probe dehydration order. Do you know what this is? FT-ICR MS? You do MS, right? Dehydration by LAN-M enzymes is known. Yeah. Elimination step is fast or hard to catch. So yeah, they mutated that. All right. Cyclotron resonance MS. Okay, so how is it different from regular MS, I guess? High resolution for MS and the most expensive. Oh, that checks out. Um, they um they do something where like they basically notice like so they want like the buildup of intermediates, and then I guess they fragment the peptide that is partially modified, and then they isolate the regions that have been modified. So does like that experiment do this? I guess. Um, 1 minute mostly starting material, and so 1p two phosphorus. Okay. If LTCM were fully processive in these, you'd likely see unmodified plus fully phosphorylated, not the latter, accumulating over time. It means the enzyme releases substrate between steps. That's unfortunate for me because simulating unbinding is very hard. Which are phosphorylated first, second, third? To use electron capture dissociation. That might be the thing that breaks it apart. C and Z ions, crucially, is a gentle, retains labile PTMs. Okay. Fragment them by ECD. To fragment the modified intermediates to isolate the positions of PTMs. Okay. So it's gentle and it retains the phosphorylations, unlike CID or CAD. They look at the C and Z ions. C50 ion is always unmodified in all states. All phosphates are C terminal to residue 50, i.e. the core. Okay. So C50 is the position. Am I getting that right? Yeah, well, this is 50, so what's a C50 ion? C terminal to residue 50, i.e. Okay, yeah, I just defined it there. The monophosphorylated, they see both unmodified C53 and C53 means sometimes residue 52 to 53 region is phosphorylated and sometimes it's not. They also see that two positional isomers, one with P and one with P. For okay, so this is for monophosphorylated species. Oh, okay, I think I understand. MSMS is to distinguish isobaric same masses when they are co-eluting, so they can distinguish them between different fragmentation patterns. ABC, XYZ ions are fragmentation patterns. You can look up by searching peptide. Thanks for helping me out on this. Oh, okay. All right, I think they have an image. Yeah, right here. C50. Man, this just looks confusing to me. I don't know. Would be the low side. I mean, here's C50. I think that corresponds to like monophosphorylated species. C50 is always unmodified and okay, so I think that's saying so anything before here was unmodified. C50 is a fragment. C53 and C53 plus 80 means sometimes residue 53 or 52 to 53 region is phosphorylated and sometimes not. What do these stars mean? Phosphorylated and then negative control. Oh. What's the negative control? Against non-phosphorylated? See those weird brackets in the sequence, those indicate the fragment ion. Okay. So going back to here. So B is anything behind that, and A, or wait, A is anything behind this. So this would be like the C alpha, right? Is that what I was talking about? So this would be, that'd be the Y. No, it points that way. It'd be the C and Z ions. C and Z. Okay. So they're looking at this. So this is, they're breaking the, this would be the peptide bond, I think. I don't know what you call this bond. Is this the peptide bond? Yeah. C and Z. So these are all C and Z's then, I believe. Are N-term cleavages. Yeah. Okay. So it goes from N to here. That's where the cleavages are. And then C term are X, Y, Z. I guess that makes sense. So early letters tell where it's cutting from, then ABC tells what position. So late letters would be, yeah, all right. Okay, so this is from end term. And then 53. Which, yeah, okay, I get it. So they're overlaying the negative controls here. What is B? The C50 ions localize all modifications to the structural region. C53 light green minor. Stars indicate in the negative control it's not treated with. Okay. So this modification here allows the buildup of intermediates. So it seems like C50 is the fragment, then serine 64 is the loci, the loci. Yeah. Alright, I don't really think I need to know this much details. Phosphorylations has been not strictly ordered. Diastereomers are isobaric isomers. I don't even, what is isobaric isomers? Okay. Um, because so the isomers would be between like a phosphorylated threonine. So, but the threonine has a methyl group in it, so I don't know why they'd be called isomers. Okay. All right. This is the one I really care about, though, Helen 2. So it looks like they did the same, but this was different because it allows for the. This one has a buildup of intermediates, whereas this one doesn't. Um, Cat is not safe because they're looking at permanently dehydrated residues, not labile phosphates. They analyze B ions, N-terminal fragments, and Y-ions, C-terminal fragments. Yeah. If the fragment contains a specific, yeah. So by checking which B ions have lost, they can tell how many dehydrations are present in the N-terminal portion. Okay. In 1 to 4 dehydration intermediates, they found that dehydrations localize sequentially, 61, 62. Positional isomers mean same sequence, but the position of the residues are shuffled. Okay, I see. Um, this is the sexy part. No, it's helping. I am confused, but it's not because of you. I just don't do this. You're helping. Um, I just need to come up with like a concise little thing. So I mean, what are they look? They're looking at the. Do they have like a T? Usually, ChatGPT writes a TLDR. Mechanistically, the paper shows that execute peptide is a distributive. All right. How do they rule out gas phase neutralizes? One worry, maybe the dehydration is coming from less water. Um, yeah, probably. Well, um, I mean, the data comes from mass spec. Like, this is kind of just background, a lot of it. Like, a lot of my data comes from HDXMS, which, to be honest, is kind of easy to follow. Like, there's no, like, weird fragmentation patterns and stuff like that. Like, here they're trying to isolate the order in which this enzyme modifies the peptide. But for me, like the most of the data I work with is just like increasing peptide mass over time, you know. So it's a lot less nuanced. And then this is like a very specific, like, application, I think, of this. I don't know. I'm not a mass spec guy. Yeah. Well, checks out. This is what I'm learning it, you know. This this is me reading the fundamentals kind of just in a specific context. Sometimes people come in here and they're like, "Man, you don't know like any of what you're reading," but like this is like why I'm reading it in the first place because I don't know it, you know. All right, so maybe I can let's come up with a concise summary of the interpretation of the MS data for HAL M2. So they observed different fragmentation patterns and changes in mass of those fragments at different time points expand upon this, please. This is only the first paper too. But I think once I understand this, then the other ones will probably be easier to understand. So this is zooming into there. 1141, so it's like around here. This is LTN 1. I don't really care about that one. Is it? Yeah. Hmm. Alright, let's see what this is. Okay, they mix at different time points, taking aliquots. Got it. Each dehydration loses water. At early times, the intact MS already shows a ladder of species. Okay. Over time, the distribution shifts towards. Okay, so that's what this is referring to, I think. Um, the precise, the presence of clean peaks for means that the enzyme is distributive. Okay. All right, so I can just copy this, I think. 7x. The presence of clean peaks. Okay. Yeah, I mean I think that's enough for me to know. Like the specifics of all this other crap. Hallam 2 seems to play nice, I guess, because it has a ladder of species. Whereas here there's like positional isomers as you were talking about. All right. Run the same reaction at quench with acid. So this will, what's the word for it? Derivatize the free cysteines. They ran the same thing but with IA. Iodo something something. Acetamide. Free cysteines on a ring cyclized cannot be alkylated. Okay. Nice. From this, each species can give peptide 6 dehydrations with IA tags. Wait, what? Oh. Okay, so they saw one ring, and then they saw 3 rings. Maybe. Compare fragment to the amount. If a B ion contains, or to the theoretical by seeing B ions at -18, -36, etc., they map the positions to the order. Do they have a graph of this? Okay, that's what this shows, I think. -18. Where's -18? Concrete examples. To see terminating region with no. All right. Oh, I see. Okay. That's just talking about the loss of water. All right. So cyclization mapping, fragments that include free carry that, yeah. Extend to carry. Cysteine 84. Hey, Parakeet. Cysteine 84. Okay. I guess they couldn't find one before then? Oh wait, never mind. No, that's 3 rings. Right. Okay. So any increase in mass means that there wasn't a ring formed. All right. I get it. Yeah. He was helping me out. I'm going to be working on this PowerPoint until probably December, honestly. To see if there are any uncyclized intermediates. They saw that Yeah, when is that again? B ions increases in mass. So that B ions in early stages have increases in mass with B ions near the N terminus head in early stages that increases in mass, and this mass decreased as time went on. This is how they know it was mostly N2C modification. Alright, I could probably go deeper, try to understand it more, but 2 p.m. Yeah, I should be able to make that. So do I just hop in the Discord at 2? Alright, that's fine. Well, I gotta write a thesis, which is essentially a book, so. Alright, let's move on to the next paper. What we got here? Discovery. So this, well, this kind of validated the other one. We should look at it anyway. So both of these were kind of validated from this paper. Biosynthesis of Alright, so what is this paper talking about? I don't know if there's like strict guidelines for the length. There's like, I think there's like 3 chapters or something like that. But usually it ends up being around like 200 pages in total. That's with like figures and sources and everything. Alright, so I think this is just talking about its discovery. They did IAA again. Oh, there's that mass spec coming back. I'll ask my professor about this technique. He can probably explain it to me in person. Yeah, 200. All right, I think we can get rid of this one actually. Um, this one's an important paper though. Substrate control and stereoselective. You get paid a lot more in your company. Yeah, this gives me motivation sometimes. Let's look. Let's look for a second. Pfizer. 135. Oh yeah, boy. All the jobs are in California or New York or Boston though. But starting out making this much is pretty good. Maybe not in California actually, but you know. This job also lends itself well to being able to operate like at home. 50 grand. Nah, that's what I'm looking at right here. This is terrible. I don't know. I think the job market will actually get better given the technologies that are coming out. All right, so let's look at this paper. Substrate, substrate control. Yeah, mainly for like career advice, honestly. Even though I think half the people on there are fake. Ooh, this is in Nature, and I don't have access to it. I don't know what to do. Where's the DOI? Oops, that ain't it. Maybe Google Scholar. There we go. All right. So before I get into this, I will do the same thing over. All right, so we're going to learn about some stereochemistry. Enzymes are typically highly sterile. We report a rare example where these natural products contain. We demonstrate that anti-additions in which the substrate determines. So I think what I'd probably need to do to supplement my rating here is pull up these rings. Where would that be? Here, probably. All right, so where's, I think it attacks this way, or maybe it's the other way. Yeah, I think it would be from here. This is the N terminus, that's the C. So cysteine is downstream, so it'll attack here. So the stereochemistry here would be 1, 2, so this would be R. And ring A is unique. So this is probably going to be S. Make my direction straight. This is N, that's C terminus. So this will be 1, yeah, so that's S. Okay. Single point mutation in the peptide substrate completely inverted the stereochemical outcome. Lipid peptides are a large family of ribosomally synthesized substances which constitutes the ground class. They are precursor peptides because of N-terminal. The latter takes place. Yeah. I know all this. Recently, it was generally assumed that cysteine addition always gave 2s6r lan, hereafter called DL lan. And 2s 3s 6r me lan, hereafter DL me lan. Figure 1b. R, yeah, that's what, and then B is S, and that's S, and that's S. Oh, nice, they have this, okay. So RR is LL. Okay. Hmm. LL. This assumption was previously challenged by the discovery of 2R, 3R, 6R me lan. LL. I already forgot. So it was mainly DL, but how M2 had LL? Is that what it's saying? Yeah. Based on the LL, we have suggested that motif. Yeah, so this is mainly, I think, tryptophan and proline, these X's here. Induces the formation of unusual, this model, the LL, is formed by anti-addition of cysteine to the reface of alkene because of the conformational preference of substrate that is unique to this motif. First, given the cystols with LL, how can one enzyme catalyze similar conjugate additions with different stereochemistries in a single polypeptide substrate? We perform LL is not specific to enzymes. Therefore, we demonstrate that D2-alanine mutation in the second position resulted in MELAN formation with canonical DL stereochemistry. So it's this second threonine here is what creates the stereochemistry. Chimeric peptides with what is a D and L amino acid? How do you I forgot how to assign D and L. So L is always like, is it S? Are they related? So L is nature's go-to stereoisomer. Designation. D is right-handed, L is left-handed. The amino acid is on the left side. Oh, the amino group is on the left side. Amino acid is the same thing. What? This is the amino group, isn't it? This is on the left side. Yeah, I mean, that makes sense. All right, so let's look at this then. So according to this paper, RR LL, so this should be, yeah, no. So this should be an L. So if we're looking at it like this, so this is the amino group, which is on the right side. So it should be D. Am I doing it wrong? I need to position it like that. Yeah. It's on the right side. Unless I parameterized it wrong. That could be. Oh no, this is ring B. Oops. Um, okay. So here we go. So the amino group is on the left side. So it's L. LLRR. Yeah. L amino acid. Okay. Let's check. So this is 1. So this is R. Yeah. So for D and L, you just have to make sure the R group is up. Really, that's the only thing you have to make sure of. Is this a this is a Carl Center as well. So this doesn't have a D or an L thing. This one does though. So this would be L as well, L amino acid. Yeah, I think so. The way you do it is just if you position the R group up like this, and then you just look at literally where the amino and carboxylate group is. So this is for ring A, which is what I'm looking at now, RR, so it should be LL. So L would be the amino groups on the left, which it is. So we're getting it. We got it. At least I'm convinced that we have it. So typically it would give DL, which is true for the B, C, and D rings. It's formed by anti-addition of cysteine to the Re phase of the alkane because of conformational preference in the substrate that is unique to this. Okay, so we probably need to look at what this is. First, I'm not used to seeing W so flat like this. What do you mean? All right. How do you assign L/D stereochemistry? That's easier than doing the R and S. You point the R group tryptophan in your ball and stick model. Usually has a bend. I've seen that in MD simulations. That's just like a geometry optimized structure. You point the R group up and then look at the amino group, and if it is on the left, then it is L, and if it is on the right, it is R. Okay. Most amino acids are L, and most cyclized products from LAN M have DL. But ring A is LL. All right. So let's look at anti-addition of C-phase. So I forgot what anti-addition means. The opposite sides of the face of a double or triple bond. Okay. So what would be the two substituents there? So here we would have a double bond normally. Mm. So it's talking about, okay. Okay, so this would be laying flat this way and this would be laying flat the other way? Or no, that's just talking about the stereochemistry after the fact. So anti-addition means, so the cysteine would come from the top and then the hydrogen would come from the bottom. So it's talking about this here. Okay, I get that, I think. What is the mode of attack for the cysteine? It is 4-ring A. To get the DL stereo, or the D assignment. Wait, so actually, I know with D and L, for cysteine it's a little bit different actually. Or no? Yeah. Wait, what? Is it different? Am I tripping? Does have two chiral centers. So here would be one chiral center. Where's the other one? I mean, you'd still do it the same way though, right? See, with here, there's no amino group, so I guess you would just, really it's talking about this, that chiral center. But this chiral center doesn't have an amino group, so you wouldn't define it as D or L? This would be R. So it'd be 1, 2, yeah, R. Whatever, I'm just not going to worry about it. So there's the paper. Okay, so anti-addition, yeah, so to get the D assignment, you need to preform an anti-addition to the double bond, meaning the hydrogen will come from the opposite direction of the cysteine attack, right? I believe that's the case. Of the reface of the alkene because of the conformational preference of the substrate. So the reface. There's re and psi. This is for my candidacy exam. And these are just papers that are just they're just background basically for the enzyme that I'm studying. Yeah. Um, what is so is the reface the top? Oh God. One, two, three. Okay. Mm. How does this work? Hallam 2 is what it's called. Generally, it's a class 2 lanthanide peptide synthetase. Use the same priority rules. Okay, that's not bad then. Basis read counterclockwise. Okay, so looking here, so if this is the double bond, there's no cysteine. So we'd look at it like there would be no proton here. So generally speaking, this is usually to the right. Yeah, I know what we could do. We could look at this. So it's going to attack here. So the re face, so this would be 1, 1, 2. So if it's, this is counterclockwise. So that is the psi. So it's going to come from here. So that'd be the re. Okay. So it's going to come, cysteine's going to come up here, and then the hydrogen's going to come down there. So this is the N to C terminus. So it would wrap around. The cysteine's going to be over here, so it's going to come, so it's going to spiral upward like that. So you can think of it like this. So it'd come this way, then the hydrogen would be that way. Yeah. The cysteine will attack from the re side. Right? From the re side to you put the highest priority up top, and if it is clockwise, it is re and vice versa for psi, I think. Okay. You really like cyclic things now. I mean, it's not about what I like, it's about what I have to do, really. But I mean, yeah, cyclic peptides are cool. There's this really interesting paper actually that came out in 2024 where they used my enzyme to modify GLP-1. And apparently the GLP-1 works better. It's more resistant to degradation to the enzymes that would typically degrade this. So could make it better Ozempic if you install rings on it. But this enzyme is what nature uses to fight against each other. I call it like the weapons factory, basically. Or it's what or bacteria use to fight against each other. I don't know if I said that. All right. These findings have raised a number. So I think they have actually a diagram here. So let's look. So this would be re-attack. So it'd be, if he had this positioned up, so it'd be 1, 2, 3. So that's re. Yeah. Well, is this not a psi face attack? Or does it come from the opposite end of what you're looking at? One, two, so that'd be re. One, this would be psi, re. Yeah. Am I assigning priority wrong? So this would be the atom. Oh no, it's this atom. Oh. So this would be one. Okay. So this would be one, two. So this would be psi. So yeah, a text from the re. Okay. That's confusing. December. It's either December 1st or like the 14th. But honestly, like this is just background. Like they might not even ask me about this stuff. So I'm just kind of doing like making sure I go in there knowing as much as I can, basically. Most of my talk is on the model that I'm creating. Yeah, I mean, I got time. I'm not worried about it, honestly. I feel good about it, frankly. One, supposed to be psi. So let's look at this. Right, so I text this atom. So one, two, psi, so a text here. One, re. So this is the n. So it's gonna come down. Or so this will be the reface. So cystine's gonna be over here and then attack this way. Okay, I kind of see why they positioned it how they did. And to see. Okay. All right, all right, all right. So without this DHB here, then it'll attack from the psi face. Interesting. Make sure you focus on the atom that E is binding to. All right. Okay, I understand that. All right, so to test the hypothesis that the substrate peptide determines the formation of independent, we generate a chimeric peptides. So they use the CYL core connected to leaders of different lantipeptides. Okay. So I guess the motivation behind this was to see if it was enzyme specific. So they put the same core attached to the different leaders. It's hydrolyzed, which were derivatized. Okay. Observed person. Hence, HELM2 catalyzed cyclization with same stereochemical selectivity as CYLM. So LA and has the canonical. almost exclusive LL and DL signals demonstrated that also preferentially forms LL stereochemistry when guided by a substrate sequence. Okay. They all have DL. Unusual. Okay. In this case, we observed an LL, relatively low intensity as well as a DL. The low signal intensity could be a result of observed incomplete dehydration. Clearly, okay. So yeah, the sequence determined the yeah, no. I wonder how long this took. Let me go to the bathroom fast. Alright, so they mutated the second threonine. Okay, the modified peptide was hydrolyzed. Yeah. Okay. Resulting amino acids were derivatized and analyzed. A DL signal is almost exclusively observed. Okay. As expected. Okay, so ring B remained unchanged. Analyze the core. So probably the core would start with DHB, which would hydrolyze to a ketone after the removal. We chose trypsin. Interesting. To investigate the possible origins. It's very rigid, as depicted in the Ramachan plots. And experiments favored synergistically to generate a stable secondary structure with characteristics of both alpha helix previously found. Okay, so the alpha helix, the secondary structure of the peptide itself encourages the rephase attack. Majority of the is also found in helix-like secondary structure, whereas the non-distorted gamma term, which is deviates from planarity due to the hydrobonding interactions. In contrast, the secondary structure sequence for psi-phase approach is more flexible. In order to approach the psi of the alkene, the reactive to forced by the rigidity of the proline backbone. This proline is an alpha. It has 70 to 30 ratio, and the adjacent is also flexible, showing two main functions. Overall, this higher flexibility and the disruption of tight hydrogen bond network suggests that the psi-phase approach might be. Okay, I don't quite understand that. So the alanine stabilized some sort of conformation more. But they talk about the proline as well. I'll ask ChatGPT about that. The planarity were confirmed. Activation barriers. Okay. A rigid oxyanion hole in this orientation, figure 5. Let's look at this. Where's the oxyanion hole? The presence of tight hydrobonding network in the reface approach, which creates a rigid oxyanion hole to stabilize the developing negative charge on the carboxyl. Okay, so it's talking about this. Okay. So in my simulations, I need to look for stuff like that, I guess. An oxyanion hole. I'm running one right now that's hopefully going to have ring A form. So we can look at this real fast. Dispatch. All right. That'll take a minute. I already have some data though. I'm just going to find it. Yeah. So let's look and see if there's any sort of oxyanion hole forming. All right. This would be res name DHA, ZDB, ZNB, CYS, CYM, ATP. Okay. All right. Well, it kind of just looks straight, honestly. So we're thinking the ring forms within the active site. may or may not be true, but I do actually have a structure for ring A formation. I gotta go in my email, I think, though. Let me go back. Let me go black real second. Oh wait, maybe it's in box actually. But ring B we know forms first is the thing. But it doesn't necessarily have to form for ring A, because there are enzymes that don't have ring B. So all right, let's look at this. All right, so this is where we think ring A might form, down up in here. So let's do our re and psi face bullshit. So if it's attacking right here, this is 1, so this would be psi, because it's counterclockwise, right? Yeah. So this is attacking in the right way. On the re psi 1, on the re side. Yeah. So that's good. Do we see this oxy anion hole that it's talking about? So it'd be this dehydrated residue in bonding to the alanine. And there's that. Do we even have an? No, we don't have an alanine. And then we can look at the residues surrounding this. So we have a histidine here, which is a very conserved histidine. There's a lysine. So all of those can act as like acid-base residues for deprotonating this. And then there'd have to be a protonation on this side, which this histidine could do. Or this aspartate. Probably the histidine though. And that would be the anti-addition. So seems pretty reasonable, honestly. And I like the secondary structures that are going on in this P50 loop. All right. So that'll be something I put in the presentation right there. All right. Where were we? You're talking about the MD stuff. Completely changes the secondary structure in both. Okay, so that's important then. In the reface approach, the reacting DHB still exhibits an alpha helical conformation, but the non-reacting and pro adopt mainly an inverse P2 conformation, planar S. What is that? Huh. PP2 helix. Bruh. Polyproline helix. Is there like a PDB with this? I don't know. Due to the release of conformational constraints imposed by the neighboring pooling and hydrodynamic ability, unlike the approach, is achieved by maintaining a reactive equal to summarizing. Defer from the samples preferentially for neutral cysteine. Notably, the lows completely reverse from those calculated. Hysteria selectivity towards LL formation was calculated for this sequence based upon the transition. It should be noted that does not allow the formation of tight. Oh, okay. Shows a normal endergonic behavior yielding a higher energy enolate. Interestingly, confers the necessary conformation bias to override the suggested less reactive. Ugh. It's just a bunch of words, bro. Just give it to me straight. Hmm. Okay, so the sp3 hybridized alpha carbon and the halonene. Yeah. So this is good because this means that to get ring A formation, you really just need to put in the dehydrated residue rather than like it be a ring formation that happens during like the phosphorylation of the second residue. I mean, I guess that could still be a possibility, but it seems like what needs to happen is after phosphorylation, the second threonine, the protein does some conformational sampling and arranges itself in a certain conformation that's allowed by the dehydrated residue. I mean, this is a cute little paper. Like, they really didn't do much experiments, did they? I mean, yeah, they did actually. All right. Um, all right, so let's talk to the ChatGPT. So, okay. So, how did they know the stereochemistry was not enzyme specific? The enzyme or peptide substrate. They show experimentally and computationally that for specific motif, the substrate sequence and its conformational preferences dictate which face of the alkene is attacked. Is this going? Yeah. So slow. It's 30%. Class 2 encyclize the canonical stereochemistry for aniline is 2s, 3s, 6r. I still don't really get that. Oh, okay, I see. So it's talking about, yeah, yeah, okay. DL emilan D for, yeah. It's the unusual L circulation something. All right. If you graph the CYL-LS core under different leader peptides, they took C corporate to the naturally contains tryptophan proline. So that's what mine has, right? Tryptophan proline, yeah. Fused it to different leader peptides. So looking at this, it seems like this can only happen when B is being cycled or dehydrated. Oh, bro, that's confusing. What is this? Spartate? Yeah. Okay. They took core peptide naturally contains. If used to different later peptides that different would recognize and process it. It's a good little summary there. Okay. So they did this again. They purified the modified precursor they used to check how many waters were lost. They saw the mass shift corresponding to 4 dehydrations as expected for. Okay. Alright, so assigned stereochemistry by hydrolysis and chiral. The modified peptide 6 molar to break all amide bonds, free amino, including LAN and ME-LAN residues, which survive as thioether-linked amino acids. Okay. It's into volatile chiral-sensitive derivatives. Now each stereoisomer becomes a different diastereomer. Ah, it's clever. Okay. Then hydrolyze the peptide. Breaking all amide bonds while leaving the rings intact. Derivatize the amino acids to volatile chiral-sensitive derivatives. So they used N-trifluoryl pseudoisopropyl esters. So now each stereoisomer becomes a different diastereomer. This paper is a lot easier to understand. And they separate them on a chiral GC column. Use GCMS with selected ion monitoring to distinguish fragment masses. Okay. If your peak coalesces with standard A, it has that stereochemistry. That's cool. GCMS here is effectively a stereochemical fingerprinting method. Okay. After hydrolysis, strong peak at retention time matching LL land peaks matching DL. So they just use proc M here. It's a very strong argument that the local sequence motif in whatever conformation it prefers is determining the fast cell selectivity of the microdisc, not peculiar features of any one enzyme. Okay. So that also suggests that it might not be zinc catalyzed then, too. And mutate after modification. GC shows a 2-to-1 ratio of DL to LL peaks consistent with shows almost exclusively DL. Um, hmm. This kind of means something that, like, it means wherever it's happening, it can be conformationally explored, basically. Like the peptide itself is not bound very tightly during ring A formation if it depends on the residue. See a mixture, the fact that it's now essentially all DL means ring A is flipped. So a single point mutation. That's so strange. Because you still need those acid and basic residues within the vicinity. This looks like a ring formation too. It's for B. This would be A. Oh, it's all so confusing. All right, I think I'm gonna take a break, and then I might come back a little bit later. So I'll see you.